For those of you who I haven't met yet, my name is Joaquin Espinosa, and I serve as the leader of the Administrative and Outreach Core for the INCLUDE DCC. And I'll speed up here to catch up a little bit and also make time for the important part of the presentation today. So for those of you who are not familiar with INCLUDE Data Coordinating Center, in 2019, the NIH called for the formation of Data Coordinating Center to accelerate the discovery of etiology and biological pathways underlying the co-occurring conditions of Down syndrome by facilitating access to and query of data from cohorts of individuals with Down syndrome. And this RFA instructed applicants to organize the proposals into an administrative and outreach core to provide program management, outreach, and education, a data management core for data collection, processing, and harmonization, and a data portal core to facilitate access to an analysis of data through an online portal. So we were very fortunate to be able to assemble a great team of collaborators shown here. Out of the SUNY kids to lead the administrative and outreach core. Then we have Dr. Brian O'Connor from Sage Bionetworks, Melissa Hendel at the University of Colorado, Robert Carroll at Vanderbilt, working on aspects of data management, and then Adam Resnick, Vincent Ferretti, Jack DiGiovanna from these institutions, taking care of the data portal. And this is a cooperative agreement with NIH. We have a project scientist, Dr. Huqing Li from NHLBI, who helped us really develop this project, and many other officers listed here and many more. So the object of our study, of course, is the co-occurring conditions of Down syndrome, as spelled in the RFA in 2019. We know now for the first time in history, we have a sizable population of adults with Down syndrome. And beyond some core phenotypes, like those shown here, now we know that people with Down syndrome are protected from some co-occurring medical conditions, yet highly predisposed to others, like those shown here on the right-hand side. So it is clear that if we advance the study of chromosome 21, we'll be able to make discoveries into many other medical conditions as well. So despite those advances, there's still much progress to be made in terms of helping people with Down syndrome. If you're born in the USA today with an extra copy of chromosome 21, you have about 50% chances of getting congenital heart disease, 10 to 20% chances of getting autism, mortality from Lyme infections more than 10-fold higher, developing autoimmune conditions more than 60%, mean age of Alzheimer's disease, which is very prevalent in Down syndrome, but also, of course, earlier at around 53 years old, mean age of death, 58 years old. So clearly the INCLUDE project could do a lot more to advance research that will elongate the lifespan and enhance the well-being of people with Down syndrome. Now, how are we going to do this? Well, I'm a big believer that individuals with Down syndrome will show us the way. Everything that I showed you so far is statistics, is a generalization about average numbers. The truth is that if you meet a lot of people with Down syndrome, you see there is a lot of diversity in this population, a lot of heterogeneity. Some of them may have all these co-occurring conditions, or many of these co-occurring conditions that are more common. Some of them may have none. And in that diversity resides the opportunity for discovery. So we like to think that a precision personalized medicine approach will clearly benefit people with Down syndrome. Now, where is this remarkable variation coming from? It could be genetic variants. It could be epigenetic mechanisms. We know, for example, monozygotic twins with Down syndrome, one of them may be born with congenital heart disease, but the other one may not. These different phenotypes may also be matched by different endotypes in the transcriptome, the protein, and metabolome, microbiome, immune profiles. Beyond the molecules, of course, there could be contributions to the environment, race, ethnicity, lifestyle, socioeconomic status, and cultural factors. Clearly, we need an orchestrated effort that will enable the testing of many alternative hypotheses quickly and efficiently. So, and that's where we come in with the Data Coordinating Center. We want to accelerate discoveries that improve and enrich the lives of people with Down syndrome through accessible data resources that enable collaboration, connected communities, and the expansion of our global Down syndrome knowledge ecosystem. This is our timeline. We were very excited within a year and a half of operations in World Down Syndrome Day 2022, March 21st of last year. We launched version 1, if you will, of the Data Hub. And I'm going to tell you more about that in a minute. We have 5 studies at launch. The national registry known as the DS Connect, this is self-reported health histories. Several data sets from the SUNY Institute Human Trisom Project, and then clinical demographics and whole genome sequence data from multiple studies listed here in a partnership through the Kids First and Include project. Later in the year, we added 2 more studies from the Alzheimer's Biomarkers Consortium in Down syndrome and from the Benaroya Institute. And as you will learn in a minute, we have many more studies now in the pipeline about to be released. This was a remarkable achievement for the field collectively. 55 terabytes of unpublished data at launch, thousands of harmonized clinical profiles, thousands of whole genome sequences, hundreds of multi-omics data sets, all ahead of publication. This is a lot of work. This is why the team has so many principal investigators and their teams. We need to not only engage the data generators, but also there's a lot of work that goes into identifying this data, ingesting it, integrating it, harmonization and modeling. And then, of course, aspects of data access, protection, sharing, governance, visualization, and analysis. We do this because we want to make sure that the Include project follows the FAIR principles every step of the way, making sure all this precious data is findable, accessible, interoperable, and reusable. And if you have been paying attention, you'll probably notice that NIH is really trying to change the culture here, a culture of data hoarding towards a culture of open science. You know, many of us were born and raised in the publish or perish culture, where we would take precious resources from taxpayers to generate a lot of expensive data that most often than not would end up in a black box somewhere, then slice that data, publish a paper in a journal that has a very expensive subscription. So the shift that NIH would like to see here is to see a rewarding of the sharing of FAIR data. And of course, we all want to publish, ideally in open access journals, but we want to make sure that the data, first and foremost, is recognized as a good outcome of our research activities. And this is all done in an open science framework. That the data is open source, that the methodology is clear, that the peer review is open, that the journals are open access, even the educational resources generated also be open to the public. So if we all do this, there'll be a lot of benefits for people with Down syndrome and for the scientists as well. Greater sample sizes and increased statistical power, more diverse cohorts, you know, by joining cohorts from different parts of the country, even perhaps internationally, be able to have bigger numbers for the study of less common co-occurring conditions. We'll be able to solve the pediatric adult divide, set up discovery and validation cohorts for greater reproducibility, then exploration before expensive experimentation, identification of available biospecimens and collaboration. So a lot of good value added from doing this type of efforts. So to wrap up here, I want to make sure that you're aware of the Include DCC website, where you can subscribe to make sure that you receive our newsletters and that you stay up to date with our activities. If you are a PI, one Include Funded grant, you have a profile. If you just got a grant, you'll get a profile soon. Please make sure to review it and make sure that you agree with how we're describing your grant and your publications. And my last slide, this is how you can participate. Continue to do great research. Please collaborate with the DCC team when approached about integrating your cohort and data sets. Promote your samples. If you have biospecimens, we have a means to advertise your biobank through the virtual biorepository. Apply for these mechanisms for data generation and data analysis. If you know of self-advocates, please encourage them to enroll in DS Connect and also prepare for a very data-intensive future. And with that, I will pass the podium to my colleagues here in the DCC. This is the agenda for the rest of the hour. We have Dr. Adam Resnick will do an overview of the portal. Pierrette will tell us a little bit about what happens, the magic that her team does to ensure data harmonization and the use of ontologies. JP will do a demo of cool features in the portal. Adam then will tell you about this really cool new thing that the portal team developed, so-called the Variant Explorer. And then Jared will take us into the realm of cloud-based analytics, and we should have plenty of time for discussion and questions. And with that, Adam, I'll pause my presentation and give you the podium.
Thanks so much, Joaquin. So let me, with that background, let me share my screen.
Will you swap this place, Adam? To
Yep.
Yeah, there you go.
Hold on a second, 2 seconds. Sorry. Sorry, for some reason, 2 seconds. There we go. So with that overview, things that, I'm really representing, as Joaquin mentioned, the data portal team. And it's within the context of what Joaquin presented that as a portal team, we really try to think and address what is a really complex landscape, for data-driven discovery through the development of a portal. And so what I'll do today is I'll briefly just provide additional context and overview of how we're thinking about the portal design and platform ecosystem, in part because we always hope to continue to engage for additional input and iteration with the stakeholder community. Now, the context for the challenge really spans the recognition that the data sources that will come into the INCLUDE program span a combination of research endeavors that have focused dedicated efforts on basic science, high impact studies that are funded by the INCLUDE program, population-based cohorts, and then ultimately also clinical trials. And these diverse sources end up creating data sets that are either resident within the INCLUDE data hub or will be resident in other contexts. And our goal as a data portal is to provide a setting for connectivity across these resources and discoverability across the setting. And this is with the recognition that these diverse data endpoints and modalities create a framework where no such single investigative solution or program can fully harness the discovery potential of any one of these data sets or endpoints. We actually require diverse cross-domain expertise and resources to address the challenge of these diverse data modalities and data resources, and that no data is really static, and that the data generation process is asynchronous, longitudinal, and doesn't occur in any one locality. And it's within that framework that we essentially have a methodology that informs the design principles of the portal. And that is that our goal is to support the either ingest and harmonization of information around clinically annotated samples, in which we then transform that data into harmonized information that can be queried and is discoverable within the portal in such a way that it can drive knowledge creation and impact. But in order to do this, and this is one of the main improvements and developments with this next generation of the portal release, we have to recognize that there are multiple endpoints for this data, and that user community at this point in time has been challenged to navigate across consortia data, multiple endpoints, multiple repositories of data in order to actually assemble the resources on behalf of the discovery framework. And importantly, these communities themselves are diverse and are thinking about multiple different data modalities, data types, and for multiple purposes in which the rest of the team will highlight in the coming few minutes. So we really think about this process of connection through design. And part of what I'll highlight briefly are things that we hope a user never actually has to think about, but in ways that we hope you recognize is required and what we've been working on empowering within the context of the data hub. Now, the traditional framework of a data hub is really a hub and spoke model. And the traditional framework of a hub is one of centralization. And there's lots of advantages to centralizing data resources in ways that standardizes that data and harmonizes that data. But the recognition is also that you cannot centralize all data. We cannot simply put all data in one location for perpetuity. And the reality of an innovation that the data portal team has really engaged is to create a framework that supports centralization of data where appropriate, but then also supports connectivity and interoperability across frameworks of data resources through cloud-based engineering and frameworks. This allows us to both bring in and harmonize clinical, phenotypic, and assay-based and sample-based data sets, represent data in a common framework and store it in a common data warehouse environment. But also at the same time, the spokes of the hub reach out and connect to other endpoints in ways that is entirely new and provides the capacity for us to represent clinical phenotypic and assay and sample-based information at other endpoints. And this is one key frameworks of innovation that we've been implementing over the last year on behalf of the Data Hub, for those of you who have been following the development. So as of this most recent release of the data portal, we now have piloted and established a federated query framework that initially supports, but can be expanded to other endpoints, the queryability of clinical and phenotypic data across multiple endpoints. So that means that essentially data can be stored in one location in the context of this setting, the Kids First data resource, as well as within the Data Hub. But as an end user, you will have a seamless experience of interacting with those endpoints and querying across that data. And this is a major milestone and a first-in-kind implementation where the Data Hub is interacting with multiple endpoints where data that might be relevant for a user's particular query can be queried across endpoints in a seamless and hopefully in a way that the user does not even know that the data resides in multiple endpoints. And this is because the Include Data Hub, as Joaquin mentioned, also implements a set of governance frameworks across multiple endpoints. And this is a key challenge for most users to interact across the accessibility and required provisioning framework of data access across controlled access data sets. This, for example, includes raw genomic data files that are managed through data access committees within the dbGaP framework, as well as open access data and registered access data in the cloud. Again, as a user, our portal team does not expect you to have to fully understand where data are. You should not actually care where data resides or in what format, but that the system itself should allow you to navigate identifying the data sets of interest and navigate the required data access frameworks, and then be uniformly empowered to both query and access those data sets. And I'll come back to that when we review the Variant Explorer later in the day. The last thing I want to highlight, because this is a continuing and developing framework for the portal, and that is that the portal really needs to serve a very wide community of data access settings. And that is users like myself who prefer sort of the point and click engagement of a portal setting to create cohorts and explore data sets. And then users, which you'll see examples of this as well today, that really would rather have access to the source files and perform deep analytics, perhaps within a command line or a specific program language within a computational setting. And we have to support that end-to-end framework of a community and allow those community stakeholders to actually interact and cross-inform a specific project. That diversity is actually enhanced by additional layers of domain-specific interest in the types of data that are represented in a portal. And this includes, as Joaquin mentioned, participant-reported data sets, clinical data sets from electronic health records, survey or clinical instrument-derived data sets, upcoming, and we'll mention at the end, imaging data, whole genome, RNA-seq, proteomics, metabolomics, and each one of these requires a domain of specificity for both harmonizing, representing, and analyzing that data in context, again, in ways that requires team-based access, use, and discoverability. A major milestone, in addition to having this underlying layer of interoperability across endpoints that is empowered by the Fast Healthcare Interoperability Resource, or this federated FHIR setting, is this emerging representation of derived data that now becomes queryable. And this is where, as Joaquin mentioned, we'll discuss a little bit about the variant search setting. Up until now, a major focus, for the portal was to really make discoverable what data are available, what data files are available, how can I create cohorts across studies, and then identify the data files that I am interested in. And with this new implementation, we are now beginning to actually surface actual derived data elements that are themselves become features that you can query and analyze, and then further define files of interest for exploration. And we'll go through that in the coming few presentations. So these are some of the portal design features and implementation that have really been new milestone of achievement cards. One is this large-scale framework for an ecosystem-based interoperability across cloud-based resources. The example being, for example, Kids First and the Include program in ways that we can then expand and implement much more broadly. And then secondarily, this additional layering of data modalities from source files to derived data, and making that derived data itself queryable in a way that's analyzable and discoverable across a governance framework of controlled access, open access, and registered tier access, which the portal navigates much more seamlessly for you as end users. And with that, I'll turn over the presentation to Paret and the rest of my colleagues for the next few presentations.
Thanks, Adam. Let me share my screen. Okay, are you seeing my screen?
We are.
Yes, we are.
Okay, great, thank you. Move my things around. Okay, so I'm Purette. I'm a biomedical data manager at Sage BioNetworks. And one of my roles with Include is working with investigators to add your demographic and clinical data to the data hub. So one of the core features of the Include Data Hub is this ability to query across data sets from multiple studies and identify participants that you're interested in from those studies, and then analyze their data together, either in Cavatica or on your own. So to enable this cohort building feature, we need to harmonize clinical and demographic data, similar to what you would do for sequencing data that's coming from different sources. So today I'll be giving a very simplified overview of the process by which we take multiple data sets that start out looking very different and then harmonize them so they can be used together in the data hub. So the top three tables here are very small examples of some of the original or raw data that we get from different studies. So imagine that these come from 3 different studies. But there are several things going on here that make it difficult to aggregate these data sets. So for example, the patient identifier columns all have different names like lab ID or patient ID or participant ID. In the race column here, all of these participants are more than one race, but that information is represented in different ways. So it's either spelled in a particular way, or there is a multiple choice where each column is a different race, or there's a list of values in one cell. And then similarly, all of these participants have celiac disease, but this is also encoded in different ways. So there's either a column for celiac disease that has true or false options, or there's, again, multiple choice where you would put a 1 if the participant has that disorder or disease, or there's like a long list of pre-text values, and celiac disease is given here as a synonym. So you would not be able to search if you were just searching on celiac disease. So in order to aggregate these disparate data sets in the hub, we've developed something called a common data model for Include. And this is shown here in the bottom table. And this data model specifies what data we want to display in the hub and how it should be formatted. So this is just a tiny piece of the data model, but some of the features that you can see here are we've developed a global identifier system that allows the Include DCC to assign persistent identifiers to participants, specimens, and data files. There are consistent terms for categorical values, variables like race. So more than one race is represented consistently here. And then we have assigned ontology terms, which I'll talk about more in a bit, to the co-occurring conditions data, so that each of these different representations of celiac disease has a standard term associated with it. So the common data model also captures information from the original data sets, like the original participant IDs, as well as how the conditions were described in the original data. And I don't have time to get into all the details, but there is documentation for the INCLUDE common data model at this link that I will paste into the chat if you're interested. Okay, so each of these study data sets is harmonized to this common data model using the R programming language to rename some of the fields, transform values as needed, and add these ontology terms. And this process is captured in scripts or notebooks so that they're reproducible, and there's a record of the transformations. So the result of this process is a harmonized data set that consists of standardized data from all of the studies. And here you're looking at a screenshot from the INCLUDE Data Hub showing the Data Exploration tab, where you can see that there are participants listed here from 5 different studies, and they are all showing consistently as more than one race. They have all of the ontology terms for all of the co-occurring conditions that they have. And then over on the left, you can search across all of the studies for other demographic characteristics or co-occurring conditions that you're interested in. And then you can also search by the participant's global ID. So I wanted to talk also briefly about these ontology terms and how we use them to power these search tools in the Data Hub. So if you click on either the diagnosis Mondo or the phenotype HPO buttons here in the search facet, you'll get a box that comes up that looks like one of these two. So first of all, what is an ontology? It's basically a standardized vocabulary that's agreed upon by domain experts and other members of a community. As my colleague Moni put it, it's a way to organize knowledge about a domain in a way that is computationally friendly. So to help with standardizing co-occurring conditions data in INCLUDE, we've chosen to use 2 ontologies, the Mondo disease ontology, which is shown on the left, and then the human phenotype ontology, or HPO, which is on the right here. So the difference between Mondo and HPO can be subtle, and there's a fair amount of overlap between the two. But essentially, Mondo has terms that describe diseases, and HPO has terms that describe phenotypic features or characteristics. So you would generally use HPO terms to describe what a clinician sees when they're looking at a patient, and Mondo terms to describe the clinician's diagnosis of that patient. And we have included the capability to search both of these ontologies to allow flexibility depending on the user's perspective on the co-occurring condition that they're interested in, and also just to make as much of the co-occurring conditions data as possible searchable between these 2 tools. So here I'm showing celiac disease, since it happens to be an example of a term that's available in both ontologies. And if you looked up, for example, the Mondo term, which I'll click on the link here. Oh, actually, I have it up here. So you'll see a page that looks like this. And within the ontology, there's a standardized spelling for the term. There's an agreed upon definition. There are synonyms, and you can see where the term falls in the hierarchy of by which the ontology is organized. So you can see the parent terms and the child terms for celiac disease. And similarly, if you were to look up the human phenotype ontology, you would see a similar page of information, just organized slightly differently. So when you're searching in the portal for participants of interest, and you want to find, for example, all of the participants that have disorders similar to celiac disease, but you want to search for a broader population, you have options to select the parent terms of celiac using these checkboxes, depending on whether you use HPO or Mondo. So in Mondo, for example, you could find participants that have malabsorption syndromes, including celiac disease. Or in HPO, you could search for participants that have abnormal intestine morphology. So you can use both options to identify the population you're looking for. So I hope this gives you a better idea of what it means when we say that we're adding new data to the hub. And the data model is still very much a work in progress. At the Include DCC, we're constantly working on improving how the model represents our current data, adding the capability to display more types of data, scaling up and automating our processes, simplifying the process for researchers to contribute their data. And then most importantly, incorporating feedback from users like all of you and other community members on how you like the data hub, how you intend to use it, what features might be on your wish list. And all of these are super useful to inform our data model. So thank you, and please feel free to contact me if you have any questions or feedback about clinical data in the hub.
Thank you, Pierrette. And I think we're going to have JP now. Do a demonstration.
Hi, everyone. My name is Jean-Philippe Sibert. I am the product owner / analyst for the Include Data Hub development portal team at Saint Justine Research Center in Montreal. So today, for my part, I'm going to be mainly focusing on the new implementations that we've done for features in the last few months within the hub, and also with a special focus on touching on the new feature, the new main feature called the variant exploration tool. And just wanted to give a highlight on this as well. So we've newly ingested 4 new studies in the production environment. So this is for a total of 11 results for the study. So we've come quite a long way from our March 2020 first deployment. So this is great. It's a nice, diverse set and rich data set, allowing for the users to have access to these resources. So this is great. So in terms of the germline variant explorer, this is the main feature here. So what is it? It's a tool that enables a search across millions of annotated germline variants directly from a performant and scalable variant database using the Include's participants' genomic files. So how this variant database is created and built is essentially using SNV variants from sequence reads that were built, that were provided by the studies, and the VCF files themselves are actually available within our file repository. So these VCF files are normalized with the clinical profiles taken from the FHIR servers that was touched on earlier in the presentation. And they're afterwards they're stored in an optimal database, essentially to have an optimal data processing and an improved data versioning management using Delta tables. And afterwards, they are enriched using the Include internal frequencies for allo frequencies, and also the external public cohort frequencies. We also include various variant annotations, so that's with DBSNFP, ClinVar, Nomad, for example. So pulling various annotations like pathogenicity, functional consequences, etc. So as you can see here on the right-hand side, the structure is very similar to what we've presented in the past with the data exploration piece. So this promotes familiarity. So essentially, clinicians and researchers can utilize the search criteria on the left-hand side. So various properties of the variant, whether that be the genomic location, consequences, as I mentioned earlier, pathogenicity, prediction scores, etc. So I'll be showing that a little bit later in the presentation. We've also developed a variant entity page, so to provide a more comprehensive and detailed information at the row level for each specific variant. And finally, for participants that are over, so if there is a variant that affects over 10 participants in our portal, so across the INCLUDE cohorts, you could redirect to the data exploration page to do further investigations of their phenotypic or diagnostic profiles and decide if you want to analyze the files associated to those participants if you have access. So in terms of the database itself, it was generated based on 3 studies, which included 955 participants, where there was an association of 74 million unique variants here. So that's really the big number here. And variants could affect multiple participants. And so that's represented here with the occurrences. So we have 5 billion occurrences. So in the page itself, we highlight the unique variants at a table result view. And in terms of upcoming data sets, we're going to continue to build and make a robust page analytical. Well, we're going to build a robust page by including a few future data sets, so with HTTP, Hackenarson, and DiskSmith. In terms of the user workflow, how it would essentially work is that users would have a specific clinical profile of interest or a genetic profile of interest. So most of the time, users have a gene, or let's say Alzheimer's, for example, they'll be looking at ApoE or APP, or if they're looking into hypothyroidism, they could look into genes like DUOX2. So essentially, you would query, you would filter across the variants, and you would identify your variants of interest. And from there, as I was mentioning, you could redirect back to the data exploration page based on that condition for the number of affected participants, or taking the whole cohort as a whole. And you could, depending on your permissions, then you could access the VCFs, GVCF, BAM, and CRAMs in the file repository to do your own analysis within the Kavatica compute environment, or even locally in your computer. So this could be used essentially for downstream analyses like functional annotations, pathway annotations with Reactome or Enricher. You could even use the variants as biomarkers to do further gene expression level measurements, just to validate if there's the variant affects the gene expression. So now I'm going to go back. I'm just going to exit this view and go to the portal. I'm going to do a quick refresher on the old, well, not old, but the features that we presented in the past. So I think you all should still see my screen. And then I'll go back a little bit to do an overview of the variants page. So very quickly, as you log on, you land on the dashboard, which is a customizable and personalizable space to facilitate data navigation across the platform. You have here the authorized studies, which is where you're going to connect to your Error Commons account in the case that it's tied to your dbGaP credentials and permissions to access controlled data files. In the case that you don't have those credentials, then there's no problem. You could access registered tier, access files, and still do analysis in Kavatica nonetheless. We also have the Kavatica projects here, but I'm going to let Jared touch a little bit more on that. But again, this Kavatica platform is our cloud compute environment where you could do further downstream analyses. Now, for the studies page, this is what I was showing earlier, we now have our 11 studies. You could do various redirects to the data exploration page. You could click here. Let's say, again, if we're talking about the permissions about dbGaP, you could click on this hyperlink for the specific studies if you want to do a request for access to their files. So this is particularly important for when you're going to be doing navigation through the variants and you have a cohort of interest. Now for the data exploration page, this is really where you're going to be doing your cohort building activities. So again, you have a summary view and tabular view here with search criteria for 3 different categories. So the participant, biospecimen, data file entities. So what's really interesting here is that the entities are actually all interconnected. So you'll see here as I'm going to be building queries that as you apply, so let's say here within one of the search criteria for participants, you could add here the Down syndrome status for trisomy 21, and you'll see here that it updated the corresponding tabs here for biospecimens and data files. So this represents the association of those entities to the specific participants in the table. So you could also have it here with this participant has 18 biospecimens and 12 files. So we also, in the participant table, we show various information on the clinical profile. Within the biospecimen criteria, we have, you could search by DNA type, for example. You could search by sample availability, and then you'll see here that this is what we call our virtual biorepository in its first phase. So essentially, you could be you'll be navigating this table and filtering using the search criteria here. At an aliquot level, you could also leverage a type of common collection event where if by clicking here, you'll see that all of these samples or aliquots, we also call them container IDs, all fall under this common collection event here on the top here, as you can see. And finally, for the data files, so again, you could search by participant ID, Pierre had mentioned that, and sample IDs, for example, create sets, save sets, but I'm not going to dive too much into the details here. And finally, for the files, you also have various metadata that you could search through. And again, the access, so if you're looking for aligned reads, for example, or if you're looking for registered tier access files, I could create a query, but you'll see the actual numbers here for the full portal. And yeah, so that provides essentially the view on the data exploration page. I did want to touch on a few new features that we've added, and this is also in a work in progress. So we have a first version of our new summary view, which is a visualization of data aggregates, where now they have a type of grid-like system where you can move them around based on your preferences. And so in the future, we're planning on having a download PNG, SVG, and TSV, so you could get information and data on the specific chart itself. So that's coming up soon in the pipeline. The other feature that we recently added was the participant entity page. So this is now, again, similar to the variant entity page. It's a comprehensive view of the specific row level participant and this is partitioned out in different sections. So you have the summary section, the profile section, the diagnosis. So you can see the whole profile of the diagnoses, the phenotypes, the associated biospecimens, for example, and the data files that are available in the portal for this participant. We also have various redirects here to the data exploration page, and you could download the clinical data for the specific participant. You could also navigate through the page itself through the anchor menu on the right-hand side. So this is a nice little page to provide a more global view on the participant. So if I go here to the data files for this participant, the other one that we recreated was the file entity page. So similar to the participant, we have various summary information on the data type, the data access, the participant and associated samples. And you could do an analysis in Cubatica if you want to copy this specific file based on your authorization. So this is a registered tier file, so I I am authorized for it. And similarly, you can redirect to the portal for that. And finally, the whole workflow is essentially you identify a specific query. So let's say I'm looking at, I'll just create a new query here. So let's say I'm looking for participants of sex male for like a specific phenotype. So let's do abnormal heart morphology. And let's use we want to leverage the information that was based on a DNA sample. And let's say we're looking into crowns, for example, so for sequencing files. And so as you can see, we filtered it down and I have access. And so this is essentially the view of the files that you'd have access to. And you could analyze and copy these files in Cubatica. So that's a general overview and for the data exploration page. Now for the variant page, we added a button here. This is a new feature. Thank you again to the team. They did amazing and hard work to get this done yesterday in preparation for the webinar. So again, this is a database and very similar in structure as the data exploration page. We have the table of results here at the bottom, the search criteria, and the query builder on top where you could save filters. So we showcase various information on the variant, the type, external public information and references with dbSNP. We highlight here the most deleterious consequence impacting the gene. However, the search criteria does apply to all the consequences of a variant. So a variant could have many consequences and can affect multiple genes. We also have annotations with ClinVar, so this is more the clinical significance, the allele frequency with NOMAD, the redirect here for the studies itself. So you could always redirect to the study for a particular variant, so you get all the information on the cohort itself in the data exploration page. And here we have the number of affected participants across all the include cohorts, the frequency, the number of alternative alleles differing from the reference genome, and the number of homozygous variants. In terms of the facets here, we have the variant type. You could always search by the locus of the variant. You have various consequences. So here, let's even start by creating our query. So let's do missense mutations. And you could search by chromosome, let's say. So just looking here, we have all the chromosomes. So let's select the chromosome 15. You could search by the position, the zygosity. We also do a calculation on the transmission based on VCF with familial information. So that covers essentially the search criteria for the variant. You could also search by gene here. So if you do, you could also upload a gene list. So I'll go with that. And as you can see, it automatically updates essentially if there's a match in our database. So you have here the corresponding ensemble gene, the DUOX2, which is a gene linked to hypothyroidism, which itself is a co-occurring condition to Down syndrome. So we could add that there, and so you'll see that the variant table updated for all the consequences of missense for this specific gene. So that's very cool. So another thing that I want to showcase. So since well, I know that DUOX2 is associated to hypothyroidism, but if you submitted it here within the gene to phenotype associations, then you'll see again that we have the 97 variants that are associated to this congenital hypothyroidism phenotype. So that's cool stuff. You could also do the opposite and search by phenotypes and not have the gene symbol here. Now I could try to create a new query. I'll go here. And so within the pathogenicity criteria, we have various prediction scores for pathogenicity. So if it's deleterious, if it's neutral, we also have the VEP impact here. And ClinVar, this is one of the more common ones where you're seeing if a variant is pathogenic or likely pathogenic. So you could add that to your query, for example, and then you could leverage some of the prediction scores here. So Splice AI is one that's new. So generally, for the most part of these prediction scores, if it's closer to 1 within the interval, it means that it's more deleterious, I think, except for SIFT and Fathom, where it's the opposite. So if it's lower, then it means it's more deleterious. In the case of Splice AI, it means that, so if we do 0.8, it means that the variant will have a high likelihood of being involved with or causing an impact on the splicing event. So if I add that here, you'll see here already that the consequences are shown here for splice acceptor, splice donor. So you can see that's where the Splice AI, which is a relatively new feature that has been available to the community. Jumping to the frequencies, we also have internal and external frequencies. Again, that I was mentioning earlier, we have publicly available frequencies that are annotated with the variants. So let's say we have NOMAD genome 3.12, which is the most recent one, and we're looking for rare variants, let's say. So that's at the 0.05. And so you'll see that it updates, and you can locate essentially the rare variants for this specific profile. And so you could also, one thing that I want to highlight as well, so you could, with the VEP impact is also another very common one. So you could use a filter for VEP high, let's say. So that means it's going to have a high likelihood of affecting the functional nature of the gene. So you could add that, let's say, and again, let's do the opposite actually, where we actually add the phenotype instead of the gene symbol. So you could have hypothyroidism here, for example. And then again, let's use Splice AI again. So let's put it greater than 0.8. So you'll see here that it updated for 59 variants. And you could see here that, again, you have the splice donors for essentially that are associated to hypothyroidism. So it's not just duo X2, there are various other genes that are affecting it. One other thing that I want to highlight here, so what I was mentioning earlier is that you could redirect to the data exploration page, essentially based on the rule that as long as there are over equal or over of 10 affected participants for a variant, then you could click here and you'll be redirected to the data exploration page. And from here, you could do essentially an analysis of their phenotypic or diagnosis profiles. And based on that, you could do the same exercise that we were doing earlier with the data exploration and identify files that you want to do for downstream analyses with and copy them to Cubatica. Now, the last thing that I want to highlight here was essentially the, let's say, it was the variant entity page. So similar to the file and participant, we did the same here. So let's just select here, and we have various information on the variant itself. So again, a lot of the annotations, general information about the properties, where it's located, the cytoband. We have the gene consequences that highlight the Splice AI score again, NOMAD PLI, so with the loss of protein function, or NOMAD LEF. And we showcase the various different transcripts from Ensemble for the specific gene consequence and variant. We have the frequencies similar that was highlighting again. So we show the ones per cohort, and then we also show the total across the 3 cohorts within the variant exploration tool. And similarly, we have the public cohorts as well. So we see the alternative alleles, the frequency, the number of homozygotes for that. In terms of the pathogenicity, we have the redirect for ClinVar, so you could click that, and then you'll see essentially what more information on that specific variant. You'll see the interpretation, the inheritance, and the condition. Here, unfortunately, it doesn't have any, but yeah. And finally, we have the gene to phenotype association. So we have the various ontologies here highlighting the conditions and how this variant may impact it, or the gene actually. And similarly, we have the anchor menu on the left-hand side. So that covers essentially what I want to showcase for the overview. Adam will provide a little bit more of a specific use case for the page. And coming back to the slides here, in terms of our next steps for the short term, we're going to be concentrating on, well, of course, testing and improving our data quality control, integrate saved sets with the variants so that you don't have to you could select a few and you could come back to them. We plan on integrating the download data feature in the Data Explorer's summary view, which is what I was mentioning earlier. We plan on fine-tuning the ETL to generate new data indices used by the portal. And in terms of the table that you just saw, we're also planning on improving it and making it more visually stimulant, and you could sort by gene and inheritance columns. In terms of the midterm, we're planning on searching by variants by participants' phenotypes. So that first category that I was showing, I forgot to show it, but essentially you could filter by study code. But we do plan on adding a few different filters, so particularly phenotypes, whether that be for HPO terms or other clinically relevant information there. We also plan on having the summary view similar to the data exploration for the variant page to get those variant data aggregations. So that'll be interesting to see from an overall global view. We also plan on including genotype quality histograms in the variant entity page. This one's going to be a lot of work, but we're planning on having a gene-centric index to display a gene view in the variant data. So essentially, you have a second tab where you have a gene view. And finally, we have the data analytics, so the pathways, the gene ontology, and gene enrichment analysis. And this is we need to have this gene-centric index to do this gene enrichment analysis. Finally, I wanted to acknowledge the various different teams. This effort was definitely not one that could be done by a single team. It definitely takes a whole village. So thank you to all the teams and not just St. Justine. I did want to take a special moment to thank the whole team because they really grinded to get everything done yesterday. So special thanks to Vincent, Evan Gerard, Adrien Paul, Céline Pelletier, Gal Altefron, Luc-Frédéric Langis, Karine Saint-Ange. So thanks again. And a special thank you to Jeremy Castanja, our technical lead at St. Justine. He's really the reason why that page exists, and he's the main architect of the ETLs in the variant database. So again, thank you to everyone. If there are any questions, feel free.
Yeah, there are a couple of questions, but before we go there, I want to extend my congratulations to you and the rest of the team. This tool clearly addresses one of the mechanisms that I posed in my introduction, right, about the role of genetic variants in the appearance or severity of different co-occurring conditions in the population with Down syndrome. And of course, before the Variant Explorer, people can push those genomes to Kabatica and run GWAS and do the types of things that Jared will share now. But for those of us like me who don't know how to do that yet, maybe I can come in and look for a specific variant that I know has been identified in the general population as, say, tracking with hypothyroidism, as you were sharing, or some other co-occurring condition, and take a look, without having to push to Kabatica large data sets. So that's super helpful and very impressive. There is a question from Mary Allen, actually, too. You said that this is right now deployed for 3 studies. And she's asking whether the UPloy controls would eventually get this as well.
Yeah, so they should be included there because we do incorporate family VCFs in there. So that includes both D21 and T21.
Great. And then I think Mary's also asking, once you generate that table with the variants and other characteristics, can you download what you get in the table?
So currently we don't yet. We do. The simple answer is that we're planning on incorporating an export as TSV at least, so that will provide a view of the specific table that you're seeing live. The issue is that it demands a lot of performance to download 57 million unique variants, a table of that much. So that's one of the complications that we're keeping in consideration. But yeah, you can save a set, and we're planning on a set of variants, and we are planning on having an export as TSV to facilitate at least a set of variants. But we can maybe take a look into having larger number of variants for the download.
Yeah. And just to add a comment, I think, Mary, I think you were also asking, just to contextualize it for JP, the variant output and the Explorer, really spans, all participants. But I think what you're also highlighting, which is a feature that I think we're exploring to incorporate, is whether you can subset them by trisomy versus disomy, within that particular table. And that's something we're currently exploring. Currently, the filtering by phenotype occurs separately, as JP mentioned, depending on the number of participants that are available to you, at least within this registered to your framework, which I'll talk about briefly, at the end.
And you just addressed, Adam, another question that Mary had. But Hui Jing has another question. JP, first of all, she's also congratulating you and the team. Do you see the question, is the CleanBar linkage manually or automatically? Is there, as the ClearBar database evolves, would you be able to?
Yes. So as part of the ETL, it's something that's really important to keep up to date. So that's definitely something that we have documents in AWS buckets where we keep up to date to the latest versions of CleanBar. I think it's done weekly, where we're updating and checking if it's done, so that it's up to date.
Very good. And I think if I'm tracking correctly, that took care of the questions. If you have a question that wasn't answered, please tag me in the chat. And with that, I believe we transition to Jared, who's going to take us into the realm of cloud-powered analytics.
Yeah, thanks so much. So hopefully Are we all good with the slides?
Yes.
Cool, thank you. Thanks so much for everyone that presented before me. Excellent job, and you really gave a good background, so I don't have to do a lot of that legwork now. My name is Jerry Rosowski. I'm a technical program manager at Valsera, and I'm going to be talking about how to use the data from the Include Data Portal and analyze it in Cavatica, which is a cloud-based platform. Before I start, I still get some questions about who is Valsera, what happened to Seven Bridges. If you're familiar with Seven Bridges, we're the company that built Cavatica and other Seven Bridges platforms. In January of this year, Valsera was formed, which is a merger of 3 companies, Seven Bridges, Perion, and Ugentic. You can learn more about going to valsera.com, or I'm happy to chat more about you outside of this webinar, what that entails. For today, I'm going to be giving an introduction to Cavatica. What is it? Why you should use it? I'll then give an example of a scientific question that we'll explore in Cavatica with a live demo. And at the end, I'll share a few resources that can help you utilize Cavatica to its fullest potential. Cavatica is a data analysis and sharing platform designed to accelerate discovery in a scalable cloud-based environment where data, results, and methods can be easily shared among the world's research community. Cavatica can be for anyone, from novice user who wants point-and-click functionality to advanced users who want to build and optimize their own analysis. We've partnered with Include for Down syndrome research and several other organizations and DCCs, including Kids First, and we regularly work with our partners to advance the technology that drives scientific discovery. At the heart of scientific discovery is data, and our data interoperability team has implemented a variety of solutions to connect with data from around the globe. As part of this effort and through our partnership with Include, we've implemented a solution that allows you easily analyze data from the Include portal on Cavatica, as JP mentioned earlier and showed you. We have similar features with other data portals like Kids First, and we also offer some data like TCGA from directly within the platform. Of course, there are many ways to bring your own data as well that you can analyze either by itself or in conjunction with other data sets. Inside the platform, you can analyze data in several ways. We have what we call Data Studio, which is really our studio and Jupyter Lab for interactive analysis and prototyping reusable scripts. We also have production level tools and workflows that you can use for scalable and repeatable analyses. And you can also build custom web apps like Shiny and Dash. Inside the platform, we have made available over 800 commonly used tools and workflows that are ready to use at the push of a button. These span many data types and modalities, and they're updated regularly and optimized for running on the cloud. Currently, all the tools and workflows in our public apps gallery are wrapped using the common workflow language. Seven Bridges is one of the founders of CWL, and our platform has been optimized to run CWL over many years. These tools can be copied and edited for your own needs, and they're also great if you're learning to wrap your own tools to use as a template, and you can compare and contrast what you've done with what's already there. Today, I'm actually going to talk more about Nextflow, which has become a very commonly used workflow language. Now that you've had an introduction to Cavatica and what it is, and you'll see the demo shortly, let's focus on a bit of science. So today I'm going to use this example of how we can do some science using Cavatica. I want to ask the question, which genes are differentially expressed in whole blood between people with trisomy and those without? To do that, I'm going to use Callisto gene expression files from the HTTP study that's on the Include Data Portal. And I'm going to use DE-seq to analyze differential expression between T21 and D21. If you're not familiar with DE-seq, it's a very commonly used package for doing differential expression of RNA-seq data. I do want to note this is not meant to be a scientifically rigorous study. It's an example for you to see how we can go from question to analysis to answer on the Cavatica platform. A typical user flow on Cavatica is going to look something like this. You'll find your data that you want on the Include Data Portal or another portal. You'll push that to Cavatica. That's going to end up in a project, which is the organizational unit in the platform. Then you'll either select or upload your own tools, do the analysis, and analyze the results. For this specific example, there are a few steps involved that I want to highlight. First, we'll grab the data from the Include portal and push to Cavatica. Next, in order to do the analysis, we need to know which samples belong to which group, so either T21 or D21. The speakers before me showed on the portal how you can find this information, but we want to make that information actionable for your analysis. And this applies to not just D21 or T21, but other Mondo or HPO terms that you might want. For this specific example, we're looking at Mondo code 000860, which represents Down syndrome. You can find this on an ontology lookup service, or actually now you can find it directly on the Include portal and choose which code you want to look at. In order to implement this, we're going to run a script that's going to scrape that information from FHIR, which previous presenters have also talked about. And then we're going to use DE-seq, which was wrapped in Nextflow, to do the analysis. I put this slide here just to make a quick point that technology is a means to answer the scientific question. I'm talking about FHIR and Nextflow, which are technologies that enable us to answer that question, but it's important to remember why, and that is we're doing this to advance science. A word about Nextflow on our Nextflow on Cavatica before I get into it. We do have basic support for Nextflow and WDL. We're actively working to implement these new features and additional support with the goal of eventually supporting CWL, Nextflow, and WDL to the fullest extent so you can focus on the science rather than the technology. If you have an extremely complex workflow now, we suggest using CWL as it's optimized for our platform and has the most support. If you do want to run Nextflow, to do that, you'll need the feature flag, and all you have to do is email support-sb@valsera.com. I've included this link, and I believe all these slides will be made available to you later, so you can see how to bring your own Nextflow app to Cavatica. With CWL, you can easily build and edit tools from inside Cavatica. Currently, we don't support that functionality for Nextflow, so you'll have to develop or use Nextflow tools on your own computer and then push those to Cavatica. There's a few steps that you can see in that docs page, but I just wanted to highlight them here. You'll have to build the script on your own, download SBPack, which is something that we've made that allows you to push your app to Cavatica. You'll have to modify a YAML file, which defines the inputs and outputs for our platform. This is not standard across Nextflow. This is specific to our platform. So I just wanted to make you aware of it if that's something that you're going to do. And then you can use SBPack to update that YAML file. And once that app is in our platform, you can use it like any other app, regardless of the workflow language. Without further ado, let's get into this live demo. I do want to mention that this is going to be a bit like a baking show and that I have materials prepared, so we don't have to sit and watch as instances spin up and things process. Because of that, you'll see me going between a few different projects, but in reality, all of this would be done in one project. I'm going to start off, as I mentioned, in the Include data portal. I'm not going to go into all the features because JP did an excellent job, but one of the things that I really like is the Save Filters feature. I can click on this and it's going to take me directly to the HTTP study with the RNA-seq gene expression data. If you go up here, you can actually look at the Down syndrome status, and we see there's 304 with T21 and 96 with D21. And then if we look at the Mondo codes, you can actually query what Mondo codes there are and how many fall into different categories. If I go over to data files, you can then select all the data files that you want and use this Analyze in Cavatica button. From here, you can either create a new project or add to an existing project. I've already made a project, so I'm going to make it easier, include webinar import demo, and all you have to do is click copy files. This will take just a minute, but once it's done, it'll give you a redirect link to Cavatica where you can see the files in your project. I'm going to skip over here because I still have to log in. Yes. Awkward silence of logging in, sorry. Okay, let's see. If it worked correctly, it'll take you to this project page. On the left side of the project homepage is a description. You can write in Markdown language. This is a great place from within your project to write down what you've done, what you're going to do, keep track. It's essentially an electronic notebook. And that way, if you come back 6 months later, you know what's in this project in terms of data, what analysis you've done, what you might want to do later. Another fantastic feature of Cavatica is the ability to collaborate with others. So on the top right, you'll see this members box, and you'll see a button that says invite new members. You can use someone's Cavatica username if they already have an account, or an email if you want them to make an account. This is a great way to add people to your project that want to work with you, see some of the work you've done, carry on an analysis that you've started and you're going to hand off to them. You can select permissions such as write, copy, execute, and admin, depending on what that person's role is and what you want them to be able to do. If I go into files, hopefully this worked. You can see all of the files that were pushed from the portal. What you'll see is that we have files and file names. And then in terms of metadata, we have this field called Fire Document Reference. This contains all the information from Fire, the metadata associated with that file. And this inside this contains the information of D21 or T21. So one of the things we need to do is use the script that we've written to pull that information. I'm actually going to jump to another project that shows how to do this, add metadata to the files. On the left, you can see I've actually populated this project with some information. The first part is about how to use this script that we've made available to get that information. There's a few pieces of information you need to run it. A Cavatica token, which you can find at the top, is essentially a way to access your Cavatica account through the API. You'll need to know which project you're working in, which you can just get at the top. And this Fire cookie essentially proves that you have authorization to open up that Fire document reference. To do this, I'm actually going to go over to Data Studio. This is where you can launch an RStudio or Jupyter Notebook. I'm not going to start a new one, but you can actually see what's already been done and the scripts that we have. So this one, you can see imports the script from GitHub, and I've made that GitHub link available to you on the slides for you to reference later. So you can use it, you can modify it, and do your analysis. If you go down, the first part is just kind of the help section. I've used this import git pass so that I can save the script and you guys can't see my API. You don't need it if it's a private project, but it's great for demos. And then the last bit is actually just running the script that's going to pull the information out of Fire. We've exported or we've printed basically every link in case there's some error. It's easier for us to access. But assuming the script runs successfully, which it has in this case, you'll end up with files that have associated metadata. So I'm going to jump to another project, which would represent what we've done after running the script. This has the same information on the left-hand side. And if you go to files, you now have files that tell you whether it's D21 or T21. So we can use those files to do analysis. I'm going to jump back quickly to the dashboard page and show you the steps involved in running this analysis. So first, we're going to decompress the files. Then we're going to create inputs for DESeq. So from the portal, you get one file for each patient or each subject. And for DESeq, we actually want one matrix that has all that information. So that's what this create DESeq input does. And then we're going to run DESeq to do differential analysis. If I go up to apps, the apps tab, I've already added the apps I need. I've run the tasks as well. But you can see what apps you've added to this project. If you want to add new apps, you can go to the top right, select new apps. You can select from public apps. You can select from other projects. You can also create your own app if that's what you want to do. If I want to run DESeq, because I've already done the prep work for it, I can click DESeq. And in the description, it tells me all about what DESeq does, what the inputs and outputs are, how it works, what the parameters are. So this is a great way to make sure you know what you're doing before you actually just point and click and get something as a result. At the top, you can then click Run, which is going to take you over to the task page, and it's going to allow you to select your inputs and select your parameters. From the left-hand side, we have some inputs that are required. It says this field is required, cannot be empty. So to show you how this works, I'm going to select, I know this one is the contrast file. So I can search for a contrast file, select, save selection, and now it shows up here. To run this, I would do the same thing. I need one for counts and one for sample sheet. These are the inputs that define how DESeq is going to run. And then there's all these app settings. Actually, it shows none of them are required, so you could theoretically leave them if you want. One that I like to do is this p-adjust method. You can determine how you're going to account for multiple analyses, and I like to use FDR or false discovery rate. You would then click run, and it'll spin up an instance and run your analysis. I've already done this for us, so I can actually go to the task page and look at this one that was completed. As you'll see, I define the inputs, I define some of the settings, and then it gives me the outputs on the other end. The work directory is great for looking at some of the logs that are associated with this task, but the files that I really want are this output files directory. So I can go in here, open up the files, and these are all the outputs from running DESeq. If you want to look at the dispersion plots that show the model that you're running, you can do that. You can save this file and put it in a presentation. The one that I'm going to look at is the results. So this shows me the statistics associated with each gene. What's the p-value and what's the significance of the difference between T21 and D21? And you'll see there's log fold change, how big the difference was, the p-value, and the adjusted p-value. From here, you can do whatever tertiary analysis you want. I've actually gone and made a volcano plot to visualize this results, which I've put into my presentation, as you might do if you're giving a presentation at a conference or if you're doing it writing a paper. You can really, from here, do whatever you need to, either with more apps and workflows or in Data Studio with interactive analysis. If I go back to the presentation I have, just to recap kind of what we did in Cubatica, we pushed data from the Include data portal, ran differential gene expression analysis with 385 samples. We did all of this. I showed it to you in less than 20 minutes. Realistically, it took me a couple hours, few hours at most, and it cost less than $6. On the right, you'll see the results. Again, this is not a rigorous study, but it shows the differential gene expression between T21 and D21. They were actually 153 genes with a false discovery rate of less than 0.05. So from there, you could look at what those genes were, maybe do some functional analysis on that, whatever you want to do. Just to kind of summarize what we do in Cubatica and why it's useful, we can quickly get from question to answer. We can easily share data and results in a secure way, and we can continue to explore questions with collaborators. And that can be PI or grad students, can be co-PIs. We have foundations and consortiums working on really anyone that you want to collaborate with. Oops, sorry about that. Moving on, I just want to give a few resources that are available to you. So I didn't talk about how billing works on our platform, mostly due to time, but actually it's not even something you need to worry about. The Include and NIH have offered this Cloud Credits pilot program, which is a great way to have Include basically pay for your analysis. So you can get pilot credits to explore the platform. But if you fall into the category of an Include user, which is someone that's using Include data, designing or developing a tool that's you intend to use with Include data, or developing a tool that can be used with Include data, you're eligible to apply for cloud credits that'll cover these costs. Initially, if you email Wei-Ching Li, she'll ask you some questions about what you're doing and make sure you fall into the right category and you're eligible. But you can get up to $1,000 of cloud credits to use on the platform. If you then want to build a bigger study and do further analysis, you can actually apply and get up to $22,000 of cloud credits. And I really can't stress enough how impactful this program is, because you can do a lot of research with $20,000. If you want to use Cubatica to do analysis, but you have no idea where to start, go to the public apps gallery and see what's there. If we don't have something, you want to build something from scratch, but you don't know how to do that, Anne Delat-Mays is a data scholar for the NIC HD, and she works very closely with Kids First and Include. I've helped her teach this class that she developed called Elements of Style, Workflow Creation and Maintenance, where you can learn best practices for building reusable workflows from scratch. There's a link here. You can actually follow along on your own and learn how to do this, build something, and then upload it to Cubatica. I also want to point out there's doc pages on our platform. So this has a wealth of knowledge about anything that you might want to do. There's a getting started, there's tutorials. If you're doing something very specific and maybe getting an error, this is where I go to for my first pass of type something in, figure out what's wrong or what documentation we already have. We also have public projects, which is kind of a screenshot on the right. If you check back here in the next week, I'm hoping to have this demo that I did as a public project, so you can go on and see exactly what I did and you can recreate it yourself. There's 2 more links at the bottom of bringing your own tools to Cavatica and bringing Nextflow apps to Cavatica. You can check those out. They really have a lot of information there. The last resource that I want to point out is office hours. Cavatica offers office hours twice a week, 10 a.m. on Tuesdays Eastern Time and 2 p.m. on Thursdays Eastern Time. But for the last 6 months or so, on the 3rd Thursday of each month at 2 p.m. Eastern, we've been doing joint office hours with Cavatica Include and Kids First. And this is a great way to come with any question that you have. Can be data related, can be portal related, can be Cavatica related. We've had people come in and ask, just tell me more about this whole thing. We don't know about it. We've also had a lot of people come in, ask questions, and then someone else on the call is able to answer, or someone else says, I also have that question. And there's a lot of collaboration that's involved there. So I really encourage you to apply for pilot credits, create a Cavatica account, start doing some analysis, and then at any point before, during, or after, please come to office hours. We would love to see you there. And with that, I'll wrap up and pass it back to Joaquin. I think actually Adam.
Thanks so much,
Yeah.
Jared. That was awesome. I modified my presentation just for the purposes of time and to review. So to recap some of these foundational milestones that are available in this current release, and as JP mentioned, this was a heroic amount of effort by this amazing, multi-talented, multi-located team for the Data Hub. But as you saw in the presentation, or hopefully as you did not see, when JP or Jared were essentially performing queries across a portal, there was no sense of whether or not they were querying, for example, a Kids First data resource that had a Down syndrome subject or a Include data set. There's seamless backend integration across these FHIR endpoints, such that data that resides across multiple locations is seamlessly integrated. As Perrette highlighted, this level of large-scale harmonization across the phenotypic setting, combined now with this large-scale ingestion of new genomic cohorts, really creates a very large tapestry of this phenotype to genotype correlation and queryability. And the way that we've harnessed it here is to allow users to begin exploring that landscape of what is now a very and growing landscape of variants in the context of the clinical phenotypic and data file setting for the portal. Now, the teams, when they present, always make it look easy, and we know that, and that's in part, why we have the office hour setting. But as you've noticed, we're really trying to span the spectrum of users, those that are interacting in the setting of a portal or a knowledge base to query, point, click, and create cohorts in ways that can then interface and partner with, talented informaticians like Jared, who can take those data files and create project spaces within a cloud environment that can then harmonize, integrate, analyze that data at scale within a portal or computational environment. And it's really that mix that is required to drive the kind of discovery that spans across these large-scale data sets that are emerging. The part that I wanted to highlight, because this is something that I think is important and something we're continuing to innovate on, is this interface between the availability of data, its queryability, and then the process that still users have to go through for controlled access setting, particularly for genomic and variant data. So we've dramatically scaled the available of genomic data, and per the genomic data sharing guidelines, read level access to data for germline variants is currently still controlled access. But the Variant Explorer, and JP highlighted this briefly, and I just wanted to briefly recap this, supports the queryability of what is available across the Include Data Hub. And this is an important first step if you are beginning as a user with a question that is gene-centric or variant-centric and are trying to assess which of the studies that the Include Data Portal and Data Hub are provisioning, you should be accessing or submitting a dbGaP a request for. So just to briefly recap to make sure that it's clear, if you are, actually, let's do the query that I think Mary was hinting at, which is if we start with a variant and we can ask the question across chromosome 21, what are all the potential variants? And more significantly, we can actually review and say, let's just look at the pathogenic or likely pathogenic variants as denoted by ClinVar across chromosome 21. And you'll see that there are actually, ironically, 21 such variants across a number of participants and studies. And as JP highlighted, in order to empower this queryability, there is a framework that essentially allows you to see which study might contain a particular variant. So this one study here, right, DS360, contains participants with that particular variant in it. But for the purposes of actually determining which participant contains that variant, you will have to go through a dbGaP access request process, and that will provide you access to the read level information for these two participants that have this particular variant within the DS360 cohort. If there are more than 10 participants, you can then actually review those set of participants within the context of the portal, and then further filter them by other criteria, for example, what type of data they might have, and then the experimental strategy. For example, there are 50 whole genome sequences for these, or files for these particular variant-containing subjects within that setting. So this is one of the first ways that we're beginning to experiment and support the queryability of the derived data sets, and allowing users to then triage which cohorts from which studies they might want to take the next step of gaining access. And once you do have that access, then you can move to the next step of not only selecting those participants, but then actually gaining access to the particular study files, and then analyzing them in Informatica, or partnering with bioinformatician like Jared to take the next step of reviewing those files and performing additional analysis within the portal. So this is the way that we hope the end-to-end framework can really complement, and this implementation of the Variant Explorer now allows users to start at the variant endpoint and go backwards into the phenotypic and file curation, whereas in the past we've shown how you can start at the file participant layer and perform a search within Informatica to reach into the variant space. So now we are beginning to go from multiple endpoints across these cohorts. The last thing I wanted to highlight is where we're heading next. So as both Joaquin and then JP highlighted, linking the biosample-based information is a key innovative framework that we're beginning to engage. And more importantly, within the Include Data Hub, beginning to explore how to represent biorepositories and biosamples within the portal that may not have yet had data generation occur on those samples, or for which you might be able to request or access additional sample to perform additional assays on those samples. We're also piloting the integration of imaging data, and we'll have some examples of this in the coming months. Importantly, within domain-specific communities that perform research, and this is very true in what is an amazingly integrated network in the downstream research community, participants participate in multiple studies. And so we recognize that there are going to be participants that are part of DS Connect, that are part of the Human Trisom Project, that are part of DS360, and that these participants have gone through the process of enrolling and consenting to multiple protocol, and multiple data sets are being generated across those participants. Over the coming months, and in the next set of work, we'll create frameworks for linking across those participants so that users who are interacting with the portal can better understand which participants have data across which studies, and really reduce that duplication of participants in ways that is connected. a lot of the harmonization-based workflows and frameworks that we established for the Include Data Hub that Perrette spoke of and that JP and Jared highlighted on the genomic front really set the Include Data Hub for a next phase of AI-ready data sets. And that is going to be another key strategic framework that we'll be engaged in. And then lastly, a key framework that we'll be engaged in is creating that longitudinal metronome of data flowing into the Include Data Hub for the same subjects. So for example, as data gets updated on DS Connect or from any of the studies, for that real world data that is longitudinal basis to be updated, intersected, and represented as linked to the data files generated from samples across these cohorts. So that describes the scope of work that on the development side for the portal that we'll be engaged in, alongside with what Joaquin mentioned, which is the continuous integration of Include data sets that are going to really grow in scale and size over the coming years. And with that, for the purpose of time, I'll end and we can engage the question and answer session further.
Thank you, Adam, and thank you, Jared, for your presentation. And yeah, we do have time on the clock for any questions or comments. And you feel free to unmute yourself or also ask in the chat. Ed, do you have a question?
Yeah.
We have one of our DS3 alumni who received intensive training in some of these topics last year. Go ahead, Ed.
Yeah, so for access to some of the files, so after you do the query, you need to get access to the files to be able to get them, if I've interpreted that correctly. Do you only have to get access through dbGaP, or are there additional access points that are needed? And also, do you have to do it for each file or the set of files that you queried? So you'd have to go through the process all over again if you had a different query? Or yeah, could you explain it a little further, please?
Yeah, I can start and then others can chime in. So those are great questions and really touch on what we currently see as an emerging setting for these multi-cohort studies. So first, the access is managed at the study level. So that means that each of the studies, once you have access to that study, you have access to all the files within that study. So your main objective as a user is to identify which studies you want access to. Once you have that access, the portal and Kvadraka will automatically know that you have access. So that the system itself constantly understands and knows which access provisioning you have and is attained. So it's not at the file level per se, but it's at the study level, from that framework.
So there's another question. Jack, do you want to take that one from Mary Ellen about the metadata?
Yeah, I could take a shot. I mean, I think there would be an option that you could transfer all of the metadata. I think what Jared didn't get a chance to show is that you can also, thinking to Perrette's demonstration, you could leverage the information in the Mondo codes or the HPO codes and decide you're going to either take all the metadata or you're going to, with the FHIR connection, make some combinations and decide, okay, anything that fits these N criteria will be classified into this type of metadata. So it really allows the researchers to personalize the metadata that they need for their scientific question and make sure that's available in Kvadraka and to the workflows that they're using.
And then just to highlight, the FHIR-based implementation for us is really a way to align to that emerging standard for clinical and phenotypic data and its exchange. Users have the options to work with the data in whatever format they'd like. And as you'll see, if you download any of the clinical or phenotypic data from the portal, those will just be tabular in nature. For us, really, FHIR is a backend exchange framework that supports a harmonization across endpoints. So that, for example, the Kids First FHIR endpoint that manages clinical phenotypic data for subjects with Down syndrome and Kids First can seamlessly interoperate with the Include FHIR endpoint because the exchange standard is essentially identical.
Very good. And Sujoy Ghosh is thinking about sharing data from all the way from India. So this is Sujoy. I don't even know what time it is in India, but thank you, Sujoy, for joining. What I'm pretty sure is a very odd time.
Thank you, Joaquin. Definitely, I will contact you regarding my data set and how can I share with this include. That's great.
Very good. Please do email me. Great. Well, if there's nothing else, thank you to all my colleagues who presented today. Thank you all for attending. And I don't want to jinx it, but there is you saw when JP was doing a demo that there were a lot of new studies coming out, and there'll be both clinical data, more whole genome sequences, and hundreds of transcriptomes, and that is coming up now in the next couple of weeks. And then we have another big batch of studies late summer, early fall. So you'll hear from us. Please subscribe to the newsletter. Every time there are new studies being loaded into the Data Hub, you will be hearing about it through the newsletter. Thank you very much, and have a great day. And
Okay, Marianne has another question.
Oh, please.
In the chat.
Yep. So Mary, a great question. You're asking if participants know their ID. So the Include portal and Data Hub operate entirely in a de-identified setting. So we do not currently essentially use IDs that a participant themselves will know, or that any user could essentially directly link to a participant. The linkage that I spoke of, and just to make sure that it's clear, is linkage between the identified IDs across studies. So, you might imagine that one study uses one ID and another study uses another ID. That linkage process is a framework we'll be developing. But for the identifiable linkage, you would still have to work with the specific study coordinator at the site performing the study in order to link and perform that kind of linkage under the approved framework of that study.
Correct. And in fact, Mary, when we work with the cohorts, we ask that the data that we be provided, whatever study IDs they provide to us, which would be transform, and there'll be another ID after that, that whatever they provide to us is 2 degrees of separation from protected health information. Some studies may give you a spreadsheet that goes 1 to 117. That's not good enough. We want to make sure that they have better coding and the identification, and we ask at least 2 steps. There'll be more about this, Mary, coming up with the application of global unique identifiers to solve the issue of potential duplicate participants, some participants that may be in 2 studies, and make sure the data is either linked or the users know that this is the same participant. We're working on it. Sounds easier than done, but it can be done, and you'll be hearing more probably later this year about our strategy for that.
Exactly.
I should point out that if anybody finds identical genomes in the Include Data Hub, do not jump to the conclusion that is the same participant twice, because I'm happy to share that we have many monozygotic twins in the Include Data Hub, both with Down syndrome. So check your premises. If you see identical genomes in the hub, it's either we have a participant that provided DNA twice, or we have a pair of monozygotic twins. Very good. So I think, yeah, that pretty much wraps it up. Thank you, everybody.